Hello everyone, I'm Yahya Fiteih. I'm a PhD student working at Dr. Villagovta's lab. Today we are talking about the cells mediating proteinase activated receptor 2 activation in the airways. So just give a quick background about part 2. Part 2 is a member of a family of 4 G protein coupled receptors, and part 2 can be activated by serine proteinases such as trypsin and mast cell tryptase. Part 2 knockout mice develop attenuated allergic airway inflammation when compared to wild-type mice. Also, interestingly, like mice overexpressing part 2 develop increased allergic airway inflammation than wild-type mice. So going to our results, we induced a bone marrow chimera model between the wild-type and part 2 knockout. This is like the results on the left side. And then we induced like ova-albumin asthma model. We found that the loss of part 2 expression in both hematopoietic cells and structural cells, which is possibly like airway epithelial cells, attenuated allergic airway inflammation. However, the loss of part 2 expression on structural cells was more significant. Okay, so since like the part 2 expression on the structural cells is important, so this is missing in the case of part 2 knockout mice, and this might be the affecting cause of causing decrease in the T helper polarization of the immune response. So trying to induce allergic inflammation in part 2 knockout, we did like another model, which is the adoptive transfer model on the right side. So what we did, we transferred already sensitized lymphocytes from wild type mice that had been sensitized with house dust mite, and then we transferred these lymphocytes to naive part 2 knockout mice and wild type mice. And then we challenged the mice with house dust mite. We found that both the naive wild type mice and the naive part 2 knockout develop airway inflammation to the same level. So now we can induce asthma in part 2 knockout with the same levels as the wild type mice. Conclusion: part 2 activation on structural cells, possibly airway epithelial cells, is critical for the development of allergic airway inflammation. However, part 2 activation on hematopoietic cells contribute to the development of the full effect. Sensitized CD4 plus T cells from wild type mice can induce airway inflammation in part 2 knockout mice with levels similar to the wild type mice. Blocking part 2 activation or neutralization of mediators released from part 2 activation might be an interesting therapeutic approach for treatment of asthma. Thank you for listening to me.